Okay, good morning, and let us start with today's class. And yesterday, actually, I could not share that detail with you. Samra, I shared it today morning. Have you checked that? I have shared you one
Yes, sir.
PDF on chromatography. So you can just
Yes, sir.
go through that because it is a total literature on chromatography. I think there are around 70, 80 slides, and it is bit in excess for you. But whatever is required, you can just go through that, and then you can use it for your studies. Okay.
Okay, sir. Okay, sir.
Okay. Can you see the slides? Yes, Samra, I have shared my presentation with you. Can you see it?
Yes, sir. I've seen the presentation.
Okay. Okay. So yesterday we started discussing about basics of chromatography. And as I have told you that chromatography, the word derived from a Greek word, chroma means color and graphene means to write. So it was discovered by a Russian botanist called, namely Dr. Mikhail Sweat. And he did his research on separation of various plant pigments. Okay. And this was one
Okay, sir.
experiment which he did. And which stationary phase Mikhail Sweat selected? It was calcium carbonate. And which mobile phase he selected? It was
Yes, sir.
butanol. So what he did, simply he took a
Yes, sir.
cylindrical column, he filled the column with the calcium carbonate, applied the paste of the leaf here, fresh leaves here, and then percolated the column with butanol. And when butanol percolated down the column, he could see some color bands. And these color bands were nothing but of different components which got separated. And that's why he named the technique as chroma means color and graphene means to write. So something like the lines have been drawn using different colored pans. So that's why the technique was called chromatography. So chromatography, there are some fundamental aspects associated with chromatography. That chromatography invariably is a biphasic system. Means there is one stationary phase and one mobile phase. So whatever may be the orientation of the stationary phase, but invariably chromatography is a biphasic system where one phase is always stagnant. It may be solid or liquid. Why it cannot be gas? Yes, Samara, why it cannot be gas? I say that it may be either solid or liquid. What is the most convenient stationary phase?
So solid is considered the most convenient.
Yes, very good. Solid is the most convenient stationary phase, followed by liquid, because we nowadays we use systems where we use liquid as a stationary phase. And when we come to mobile phase, two state of matter they are used as mobile phase, either liquid or gas. So which one is the most convenient mobile phase?
So liquid one.
Liquid. Why not gas? Which one is the ideal mobile phase?
Sir, the liquid phase is the ideal one.
Ideal one is actually gas, because gas is
Okay, sir.
something which is having a property to be mobile, right? Liquid,
Yes, sir.
like you have to make it mobile, but gas is itself, is having a
It's simple.
property
Yes,
which
sir.
itself is a mobile. So gas is
Yes,
one
sir.
of the best mobile phase, but it is not very convenient, because to make a system from gas, it need to, like, you need to flow it from a region of high concentration to a region of low concentration. That means you
Yes, sir.
require a system for that. But liquid is said to be the most convenient mobile phase, because easily you can flow the liquids. Okay, so that's why liquid is
Yes, sir.
considered as a convenient mobile phase.
Convenient.
Okay,
Yes, sir.
right. So
Yes, sir.
this is about classification of chromatography, because so many variants of chromatography are available because it is one of the most versatile technique available in the hands of either biologist or a chemist. So there are certain classification of chromatography. So based on mobile phase, first classification is based on mobile phase. So there can be three kind of mobile phases. Two mobile phases we have just discussed. One is liquid as a mobile phase, another is gas as a mobile phase.
Yes.
And the third one is called supercritical fluid chromatography, where the mobile phase is a supercritical fluid. It is a new kind of technique that has been introduced few years back only, and it is widely used in certain kind of application we will discuss. So when we say liquid chromatography, what does it mean?
I mean, means the mobile phase is liquid
Mobile phase
state.
is liquid. Always keep it in mind. Stationary phase may be anything. Stationary phase may be solid, stationary phase may be liquid, but the mobile phase here is liquid. And gas chromatography, when we say, then we say that the mobile phase is gas.
Yes.
Supercritical fluid chromatography, in mobile phase we use a supercritical fluid. Okay, I will explain it. Okay, then the second classification is based on the principle of separation. So based on principle of separation, chromatography can be divided in 5 heads, broadly 4. The first class principle is based on polarity. Based on polarity, and based on polarity, it is divided in 2 heads. One is called absorption chromatography, and another is called partition chromatography. So I think, Samara, you might have heard about this word absorption. What is that?
Hello.
Yes, Samra, have you heard about the word adsorption chromatography? Adsorption, have you heard about the word adsorption?
Yes, sir.
What is that?
Yes, sir. Sir, adsorption means the attaining of certain liquids from on the surface of the phase.
For example?
And so it's the common example that if we dip a chalk in the ink, then it is considered as adsorbed, not the absorbed one.
Okay, and can you give some example of absorption? Can you differentiate between adsorption and absorption?
Sir, absorption, we can say like it dipping a sponge in water or something, that it just get the liquid phase just get inside of the sponge. The inside molecules of the sponge absorbs the water molecules.
Okay, okay, you're partially correct. When we say adsorption, nothing is going inside. Everything is retained on the surface.
On
like
the surface.
You take suppose you take a ball and paint it red. That means the red color paint is on the surface. It is not going inside. So this is called adsorption. And the example you gave, like you to take a sponge and then put it in water, so water is going inside the sponge. There, like on the surface, when it is there, it is adsorption, but when it is going inside, it is absorption. Okay, so in adsorption chromatography, one truth is there, and the truth is means the molecules of the sample, they are adsorbed or they are retained on the surface. So when they are retained on the surface, that means that there is no space to go inside, right? Like for absorption, there should be space where molecule can go inside. So for adsorption chromatography, the particle of the stationary phase are spherical in nature. Are spherical in nature. Spherical means what is the difference between circle and sphere?
Yes, sir.
Tell me what is the difference.
Yes, sir. Circle is the sir. Circle is considered the 2D phenomena, and the spherical is a 3D one.
Oh, again, you just explain. I could not get it properly. When we say sphere, sphere means it is solid from inside. Sphere means it is solid from inside, just like a
solid from inside.
And when we say circle, so circle is hollow from inside, just hollow. Like in a table tennis ball or a lawn tennis ball. Okay.
Okay, sir.
Okay.
Okay, sir.
So when we say adsorption chromatography, that means the stationary phase particle is solid. It is solid from inside.
Okay, sir.
That means the stationary phase is always what kind of nature? It is always solid. We discussed now which two states of matter can be stationary phase.
Stationary phase. The liquid and the solid one.
The liquid and the solid one. Very good. So in adsorption, what will be the nature of the stationary phase?
The solid phase.
It's always solid. Very good. You should remember it. In case of
Okay, sir.
adsorption chromatography, the stationary phase is always solid.
Solid
That
one.
means it is spherical. So anything which can be retained or it can only be retained on the surface. It cannot go inside the molecule, inside the stationary phase particle. Okay.
Okay, sir.
Second is, and it is based on polarity. And how it is based on polarity? It's very simple concept. Like if you have a stationary phase particle which is having a polar surface, then polar molecule of the sample, they will adsorb more, and non-polar molecule will adsorb less or will not adsorb at all. Okay, because simply it is a polarity based phenomena, and possibly you know that in polarity we call it that what do you call it, like dissolves like. Polar dissolves polar and non-polar dissolves non-polar.
Non-polar.
If the solid stationary phase is polar in nature, polar will be retained. Polar will be retained. I'll just explain it, how it happens actually.
Okay, sir.
So suppose if I explain this concept and if I just show you this picture, and if I say that the stationary phase is a solid, it's a solid and polar. Now, can you predict the polarity of these three compounds, red, green, and blue? I'm saying that the stationary phase is polar and mobile phase is say non-polar. So can you just explain the polarity of these three compounds? Can you predict? Just try to predict.
Sir, I don't know.
Just try to predict.
Okay.
Like we said that in case of adsorption, polar dissolves polar, means polar will be attracted more and non-polar component of the sample will be attracted less. So if the component of the sample which is attracted more or which is attaching more or which is retained more on the solid surface, its movement will be fast or slow?
Sir, its movement will be fast.
How come it can be fast?
No, sir, it can be slow because it is just retained on the surface.
If it is retained on the surface, because the stationary phase tend to become stagnant. So anything which is having more attachment on the solid surface tend to become stagnant. Tend to become stagnant.
Stagnant, yes, sir.
And the molecule which are retained less, because as we said that it is a biphasic system. Okay, and in biphasic system, okay, since it is a biphasic system, it is means there are only two phases. One is stationary phase and another is
mobile phase.
Mobile phase. So the molecules are having only two options. So there are only two options. The component of the sample, either they may go to the stationary phase or they may go to the mobile phase. Now, if the stationary phase is polar, then polar compound will go to the stationary phase, and non-polar components, they will be forced to stay in the mobile phase. Okay. So anything which is moving fast is something which is moving with the speed of the mobile phase, anything which is slow, that means it is having strong interaction with the stationary phase. So just try to understand the principle. So if in this three component mixture, you can say that which one will be the slowest. Which one is the slowest in this three component mixture? You can see that is red, green, and blue. Which one is moving slow?
Sir, the one whose
Just tell me the
absorption
color.
potential is more.
Already you have your colors on the screen. Now you have red, green, and blue. So which one
Sir,
is
I am
moving
not able
slowest?
to see it.
What you are not able to see?
So you have shared the screen, but I can see only the
I again shared it. Can you see it now?
yes, sir. Now I can see it. Yes, sir.
You have to inform me, na? Because due to network connectivity issue, sometime like we cannot. I am moving the screen, but sometime it is not moving at your end. Okay, can you see it now? So there are three colors in the mixture. It is red, green, and blue. So which one is moving slowest? Yes, Samra, can you see me? Can you hear me? Yes, Samra, can you hear me?
Yes, sir. I can hear you.
Okay, can you see the screen where three component mixture is being displayed?
No, sir. Not yet.
Can you see it now?
Yes, sir. Now I can see the screen.
You can see the screen, and there are three colors of a
Yes, sir.
component of a mixture. So you
Yes,
have
sir.
which colors you can see?
Yes, sir. The blue, green, and red one.
Blue, green, and red one. Eh? Okay, now tell me that which one is moving fast and which one is moving slowest.
Sir, the red one is moving fastest because it has the lowest absorption potential. It is not
Okay.
retained in the solid phase, and the
Okay.
blue one is
Now,
the
if
highest
very
absorption
good,
potential.
very good. Now, if I give you one more input, that the stationary phase is say calcium carbonate, which is polar in nature. It's like Michael-Sweat's experiment, and the mobile phase is
Yes, sir.
butanol, which is non-polar in nature. Now, can you predict the polarity of all these three different colored components?
Okay, so the blue one is the most polar because it just gets more attracted towards the polar phase, which is the calcium carbonate one, and the red one is the least polar.
Very good, very good. So we were not having any information about the polarity of the components, but still by doing one simple experiment we can find out that what is the nature of the polarity of these components. Okay, you got it?
Okay, sir. Yes, sir.
So it indicates that the separation is based on polarity. Okay, now I moved to the next slide, the classification slide. Can you see it?
Yes, sir.
Now classification of chromatography slide is visible. Huh?
Yes, sir.
So based on polarity, we have two kind of chromatography. One is adsorption, in which the stationary phase is always
Polar.
No,
Solid
always
phase,
solid.
the spherical one.
Always solid.
Yes, sir.
Always solid. It may be polar or it may be non-polar also.
Okay,
But
sir.
it should be always solid in nature.
Solid one.
That means
Okay, sir.
the retention is on the surface, not inside the molecule. Okay.
Yes, sir. Yes, sir.
Second chromatography based on polarity is partition chromatography, in which stationary phase is a loose matrix. It is not a solid matrix. So partition chromatography, the idea of partition chromatography came when we invented liquid stationary phases. When we invented liquid stationary phases, because earlier the earlier discovered stationary phases were solid because they were the convenient stationary phases. A solid can be easily made a stationary phase. But how to make liquid as stationary phase? Do you have some idea how to make liquid as stationary phase? Do you have some idea how to make liquid as stationary phase?
No, sir.
So we have a concept called bound liquids, and you might be knowing that there are so many molecules or particles on which we always have bound liquid. Like you simply do an experiment in lab, that you go to garden, take the driest soil, which you consider that it is the very dry soil and it is not having any kind of say moisture, and simply put it in a china dish and keep one inverted watch glass on the china dish, and you'll find the droplets of water. So since you considered that the soil was totally dry, from where did you got these droplets of water? Some idea?
Yes,
This,
sir.
this was bound water actually. This was
Yes, sir. Yes,
bound
sir.
water. So this
Bound water.
concept
Yes,
came
sir.
at a later stage, that we may make liquid as a stationary phase, and we can bound liquid to any solid material. Okay, and then we can use it as a stationary phase. So why we invented liquid stationary phases? Because one advantage with adsorption, disadvantage with adsorption was because adsorption is a strong phenomena. Strong phenomena means, suppose if I have 100 molecules of the sample, then maybe some 20-30% of the molecules, they are permanently adsorbed on the solid surface. Means these
Okay,
molecules
sir.
can never be taken out because adsorption is a strong phenomena.
Strong phenomena.
I
Okay,
want
sir.
to weaken this phenomena. I want to have interaction of the compound with the stationary phase because that is required for separation. But I want that interaction to be little bit weak. So how it can be made weak? I cannot change the nature of the sample because that is universal. I cannot change it. But yes, I can change the nature of the stationary phase.
Stationary
So
phase.
in
Okay, sir.
changing the nature of the stationary phase, one idea was that we can convert it from solid to liquid. And if we convert it from solid to liquid, then definitely it will be having a lesser interaction. The interaction of the stationary phase with that of the sample molecule will be decreased, so that
Okay,
we
sir.
will have separation, but we will not have unwanted retention or strong retention of the sample particles. Okay.
Okay, sir.
So for doing this, it is like for doing this, what we have done, we have made liquid stationary phases. So that means now you have liquid as a mobile phase and liquid as a stationary phase, or you have gas as a mobile phase and liquid as a stationary. So in partition
Stationary phase. Okay, sir.
chromatography, the stationary phase is always
Liquid.
liquid. In adsorption chromatography, the stationary phase is always
Solid.
solid. Very good. And polarity may be anything. Even the solid stationary
Okay,
phase
sir.
can be polar or non-polar. The liquid stationary phase can also be polar or non-polar, right?
Polar or not polar. Okay, sir.
Okay.
Okay, sir.
So this is separation is based on polarity, and in case of partition, we get separation, but the separation was faster as compared to adsorption, because the interaction of the compound with the stationary phase was weak as compared to adsorption. Okay, so this was second principle. In third principle,
Okay, sir.
we got a principle that is called size exclusion. Like in first these two principles, the separation was based on polarity. Separation was based on polarity. Okay. Now,
Polarity.
polarity means like if stationary phase is polar, polar compound will move slow. If stationary phase is non-polar, polar compound will move fast. So that was one principle. Okay. You understood it, I think.
Yes, sir.
Okay. Now, for some separations, like suppose I want to separate protein. Proteins are very complex molecule. So simply based on polarity, it was very difficult to separate those molecules, right? Because these are not simple
Yes, sir.
few Dalton molecular weight compounds. There are like very big molecules of say 100 kDa, 200 kDa, like this, big proteins. So for that, like polarity was very difficult to assess. So we came across the idea of separation based on size. So if you have to separate a molecule based on size, invariably one change we have to make it in the stationary phase. What change first? What change would you propose to make in the stationary phase?
So the stationary phase
From your
should not be
polarity,
solid.
solid. Yes, one change is there that it should not be
Yes, sir.
solid because, like, because the one first change that you will make that it should be inert. Because
Okay,
our
sir.
idea was that we do not want the polarity concept in separation. We want the size
Okay,
concept in
sir.
separation. So first thing what we did, we made the stationary phase inert. When we made the stationary phase inert, that means now the interaction of the sample molecule with the stationary phase is not on the basis of polarity, right?
Yes, sir.
Not on the basis of polarity. It is on the basis of size. Now, how we can do
Size.
it? That second change we have to made, that the stationary phase must be porous, like a liquid. It may not be liquid,
Yes, sir.
but it must be porous. It must be
Porous.
porous.
Okay, sir.
So instead of using this spherical stationary phases, we started using the hollow stationary phases which are porous in nature. Okay.
Okay, sir.
So when we created pores in the stationary phase, we regulated the size of the pores. Okay, we regulated the size of the pores. This I will explain you so that the separation can be done based on the size. One size can be excluded and one size can be included, and beyond the like that concept we get separation. Okay. Now the fourth principle was ion exchange. Again, like it was used for separation of molecules which are ionic, particularly the protein. Because proteins, you might be knowing that there are either anionic protein or cationic protein. So based on the nature of the protein, like suppose I have a protein mixture, but I am from that mixture, I'm only interested, my protein of interest is anionic in nature. Okay. So that means I can easily separate this kind of protein or all anionic protein from rest of the protein, because rest I do not require. So in ion exchange chromatography, what change we made it in the stationary phase?
We can make the stationary phase charged.
Yes, very good. So we made the stationary phase charged. So either the stationary phase was having positive charge or it was having the negative charge.
Negative
So
charge.
if the
Yes, sir.
stationary phase was having positive charge, all the anionic proteins, they bound to it, and all positively charged proteins are flushed out. Suppose my interest is only in the negatively charged anionic proteins, so I can make the stationary phase positive, so that rest of the proteins are flushed out and only the protein of my interest are like adsorbed, so that I can filter out the unwanted protein from the mixture. Got it? You got the idea?
Yes,
So
sir.
this was one like pre-separation kind of concept where it can be done very easily. And next principle is based on affinity. affinity chromatography. This is one of the most, say we can say that is specialized kind of chromatography, in which what we do, like it is affinity based separation. So affinity is like affinity of a substrate with the enzyme. Okay. Now suppose I want to
Okay, sir.
separate an enzyme from a cell sap. So simply what I will do in the stationary phase, I will take its substrate, that will be the ligand, and that substrate will be attached in the stationary phase. So what will happen? Out of the protein mixture, only the specific protein will attach to its ligand, and rest of the proteins will be flushed out, right? So this kind of separation is based on affinity,
Yes,
and
sir.
it is highly specific. So if I want to separate a particular antibody from a mixture, I can have ligand will be what? The ligand will be,
So we can have the like the antigen.
yes, very good. The specific antigen will be the ligand. So a particular protein will attach to it, and rest of the protein will be flushed out. And then later on, by changing the concentration of the buffer, I can easily take out the protein of my interest. So I can concentrate and I can purify the interest biomolecule. Okay, so you got it. So far we
Okay, sir.
discussed about two classification. One is based on mobile phase, another is based on principle. I hope you're able to understand it clearly.
Yes, sir.
Now moving to next slide. Uh, there is another classification that is based on the orientation of stationary phase. That is based on the orientation of stationary phase. So the stationary phase may either be oriented or oriented on a plane surface, plane two-dimensional surface. Okay, so if the stationary phase is applied
Yes,
on
sir.
a plane two-dimensional surface, then it is called planar chromatography. I think you might have seen the TLC plates. Have you seen TLC plates?
Yes, sir.
Okay, so TLC means when the chromatography is planar, when the chromatography is planar in nature. Okay,
Yes, sir.
then
Yes, sir.
or it may be oriented inside a column, like you have seen that what Michael Sweat invented was what kind of chromatography? It was column chromatography.
Column chromatography. Yes, sir.
And later on, like nowadays, we have some fast chromatographies which are, say, our what you call it, our planar chromatography, which are very fast as compared to column chromatography, and these are very good for analytical purposes. Okay, then
Okay, sir.
based on the polarity of stationary phase, like in one chromatography, we discussed about the like whether it was adsorption or partition. It was nature of stationary phase, solid or liquid. Okay. Now, based on polarity of stationary phase, chromatography is divided in two heads. One is called normal phase chromatography, and another is called reverse phase chromatography. Okay. So you can write down that in normal phase chromatography, the stationary phase is polar in nature. The stationary phase is polar in nature. And mobile phase, we start with non-polar mobile phase, and gradually we increase the polarity of the mobile phase. So mobile phase, we start with non-polar and we go towards polar. Okay.
Yes, sir.
In case of normal, and what Michael Sweat discovered, it was a normal phase chromatography, because he used calcium carbonate as a stationary phase, which is polar in nature, and he used butanol as a mobile phase, which is non-polar in nature. Okay. Then
Yes, sir.
we got a chromatography called reverse phase chromatography. So in reverse phase chromatography, the stationary phase is non-polar in nature. The stationary phase is non-polar in nature. Okay. So how it can be done actually? How it can be done? How it can be done? Like you can have a stationary phase which is non-polar in nature. Okay. And how? Why it was
Mm-hmm.
required actually? So suppose you think about that Michael Sweat started with the calcium carbonate as the mobile phase, as the stationary phase, and he's selected what? butanol as a mobile phase. Okay.
Mobile phase. Yes, sir.
Nowadays, like suppose, like either Michael Sweat was like very wise, or he has hide something. Like tell you, like if I tell you, if I ask you that which is the most common liquid available around you,
Water.
water is available. Now he was having calcium carbonate as a stationary phase. So why he didn't used water as a mobile phase? And if he would have used water as a mobile phase, what would have been the problem? Now, can you explain? Yes, Samra, can you explain? Just think of, just think of. Because water was the most convenient, most easily available solvent. So why he didn't use water? Okay, I give you the answer. Huh? You can hear me now?
Okay, sir. Yes, sir.
Okay, he might have used water, but if you use calcium carbonate as a stationary phase and water as a mobile phase, then calcium carbonate will dissolve the calcium, like water will dissolve the calcium carbonate. So if water will
Yes, sir.
dissolve the calcium carbonate, then what you will have? What problem you will have? Problem will be that you will have a uniphasic system. Now you have a solution
Yes,
or
sir.
you have a slurry. You do not have two distinct phases, stationary as well as mobile.
Mobile phase.
That was a reason that Michael Sweat used a stationary, a mobile phase, which was like the two systems were immiscible. So that is the first requirement of chromatography, that you should have
Yes,
two
sir.
distinct phases. One is stationary and another is mobile. So if our
Yes, sir.
mobile phase is dissolving the stationary phase, then we do not have a biphasic system. And if we do not have a biphasic system, we are not doing chromatography. We are doing something else, but we are not doing chromatography.
Yes, sir.
Okay. So when he
Okay, sir.
started with calcium carbonate as a mobile phase, as a stationary phase, he selected a non-polar solvent, and possibly he might have selected this non-polar solvent after series of failures. He might have used water, but he didn't report. It is nowhere reported that
Yes, sir.
Michael Sweat used water. And in my opinion, he must have used it in first go. He must have used water only, but his experiment
Yes,
would
sir.
have failed. But nowadays,
Yes, sir.
we have certain stationary phases, solid stationary phases, which are not even soluble in water. So you noted down that the most common solid stationary phase that is used today in chromatography, it is silica gel. You might have heard about it. If not, then you write it, silica. So silica is basically, it is obtained from diatomaceous earth, and silica is largely used as a solid stationary phase nowadays.
Solid
Okay.
stationary phase.
And it is polar in
Yes.
nature. It is polar in nature. Now, one advantage with
Okay.
silica is that it is not soluble in any of the solvent available on this universe, because silica is polar in nature. So what kind of solvent can dissolve it? Polar or non-polar?
The polar one.
Polar one, very good. But incidentally, it is insoluble even in the most polar solvent, water. So it is one of, you can note it down, that it is one of the most widely used stationary phase today, which cannot be dissolved by non-polar as well as polar solvent. So if I want to use
Okay, sir.
water as a mobile phase on silica, I can use it, right? Although
Okay, sir.
separation is little bit difficult because you have a highly polar stationary phase and a highly polar mobile phase. So the interaction becomes so strong that the separation becomes very poor. But yes, water can be used as a mixture with other mobile phases like methanol or like ethanol, which are polar solvents. Okay, you got it?
Okay, sir.
Okay, now but
Yes, sir.
problem came that when we are using silica as a stationary phase, so we cannot use water as a mobile phase. Why we cannot use water as a mobile phase? Because water is highly polar. So water will be having highest interaction with the stationary phase. If you remember, what is the molecular weight of water? What is molecular weight? What is formula of water? Molecular formula of water. Yes, Samra, what is molecular formula of water? Yes, Samra, can you hear me? Yes, Samra, can you hear me?
Yes, sir. Yes, sir.
So what is the molecular formula of water?
Molecular formula H2O.
H2O, very good. And what is the molecular weight of water?
Molecular weight of water.
Just tell me. You can just think and you will be able to tell it to me. Molecular weight of water is 18.
H2O.
18,
18. Yes, sir.
Okay.
Yes, sir.
Now you think of, I say that water is highly polar. Why I say that, what you mean that why water is highly polar? Why water is highly polar? If you compare a molecule like say glucose is polar or non-polar?
Glucose.
You might have taken glucose in your life many a times.
Yes, sir. Yes,
So
sir.
how you have taken glucose?
And water.
In water. So
Yes, sir.
is it soluble in water or insoluble in water?
Yes, sir. It's soluble in water.
And if it
It's
is
polar.
soluble in water, it is polar or non-polar?
Polar.
It is polar. Very good. Eh? Now, which one is more polar? Water is more polar or glucose is more polar?
So water.
Okay. Why? Your answer is correct. Explain. Yes. Are you able to explain it?
No, sir.
Actually, if you see the structure of water, there are two covalent bonds between oxygen and hydrogen,
Okay, sir.
two hydrogen.
Yes, sir. Yes, sir.
And both the covalent bonds are highly polar. So it's we have two dipoles, right?
Yes, sir.
If you
Yes, sir.
remember
Yes, sir.
the structure of the glucose, what is the backbone? What is in the backbone of the structure of glucose? How many carbon we have in glucose?
6.
6 carbon. So 6 carbon, they are in a linear chain. So we have
Yes, sir.
5 carbon-carbon bonds. Now all
Yes, sir.
carbon-carbon bonds are what kind of bond? These are covalent or non-covalent bonds?
So they are covalent.
Covalent bond, but these are covalent polar or covalent non-polar?
So covalent polar.
No, covalent non-polar. Because whenever
Non-polar.
there is
Okay,
a
sir.
bond between two homoatomic atoms like carbon-carbon, it is a perfect non-polar bond, right?
Okay, sir.
So
Okay,
all
sir.
carbon-carbon bonds are non-polar. All
Okay, sir.
carbon-hydrogen bonds are again said to be non-polar. Now the only polar
Okay, sir.
molecule in glucose is, what is the polar molecule in glucose? Polar atom in the glucose. What is the polar atom in glucose? What is the molecular formula of glucose?
C6H12O6.
O6. Very good. Now, which one is the polar atom in glucose? You know, there are oxygen is present in which form in glucose?
Yes, sir. Yes, sir.
Tell me, oxygen is present in which form? In which functional group?
Sir, hydroxyl.
Yes, very good. One is hydroxyl. There are two more combinations. Okay, so one combination is hydroxyl. You might have seen that carbon is bonded to hydroxyl. And when carbon make a bond with hydroxyl, what is full form of, what is the formula of hydroxyl group?
OH.
OH, very good. So when we have a COH bond, then always this bond is polar or non-polar?
Polar.
Polar bond, very good. So always this bond
Okay,
is
sir.
polar. Now two more combination are there. One is ketone, where carbon is double bonded to oxygen.
Okay, sir. Yes, sir.
Right?
Yes, sir.
Again, this bond is polar.
Yes, sir.
And then we
Polar.
have the
Okay, sir.
aldehyde combination. Right?
Yes, sir.
You have the
Yes, sir.
aldehyde
Yes, sir.
combination, and again this bond is polar. Right? So,
Okay, sir.
like if you see the structure of carbon, then only few bonds are polar,
Polar.
but maximum bonds are
Yes, sir.
non-polar.
Sir, non-polar. Okay, okay.
That's why it is polar, but it is not as polar as water.
As water. Okay, sir.
Because in water there are only two bonds, and both bonds are covalent polar bonds. So that's why it is highly polar. Okay. So when we started using, say, silica for separation, if you separate, say like your chlorophyll on silica, it is a very good combination, because chlorophyll molecules are non-polar in nature. You have silica as polar, and you can always use a non-polar solvent like chloroform, petroleum ether, or hexane as a mobile phase on this
Non-mobile phase. Yes,
chromatography.
sir.
Okay. But if you want to use protein, if you want to separate, then what problem will come? Because although protein may be polar also, but when you put water in protein, then what will happen? Your water is more polar than protein. Why it is so? Because water is having two bonds, or and both are strong dipoles. So it is the most polar solvent available, right?
Yes, sir.
The
Yes,
water
sir.
molecule will interact with silica, and your protein will not be able to interact with silica. So if there will not be any interaction with the protein with silica, that means there is no retention, and if there is no retention, there is no separation. Okay.
Okay,
So
sir.
if I want to separate protein on silica, either I have to use organic solvent. Now, can I use methanol or chloroform as an organic solvent when I'm separating proteins? With your basic knowledge about the biochemistry, can I put protein in an organic solvent? Yes or no?
So we can use it.
Yes.
Hello, sir. Oh, hello, sir. Hear me?
Yes. Tell me.
Yes, repeat the question. I'm not able to hear you properly.
Okay, I am saying that can we put protein in an organic solvent like chloroform, petroleum ether, or methanol? The answer is no. Okay. Yes, Samra, can you hear me? Yes, Samra. Yes, Samra.
Yes, sir.
So my question was, can you put a protein in organic solvent like hexane, chloroform, or methanol?
So we can use it depending upon the polarity. That if the protein is polar,
Okay.
then
And the
how
answer
it will
is
elute.
no. Why no? Because protein is only stable in water. Okay. Protein is
Okay, sir.
only stable in
Stable
water.
in water.
Or protein is not stable in any kind of organic solvent. If I put it in the organic solvent, then it will simply precipitate. It will be a non-functional entity, right?
Okay, sir. Yes, sir.
So if I have to use protein, my mobile phase should always be
Water.
very good. It should always be water. And if I am using silica as a stationary phase, my mobile phase cannot be water. My mobile phase cannot be water. So there was a tricky
Cannot be water.
situation. Because why it cannot be water? Because water being highly polar will have a very strong interaction with silica and it will not
Silica.
allow
Okay.
anything to separate.
Okay, sir.
So on silica, we need to use organic solvents, but for proteins, we cannot use organic solvent. So that
Okay,
was a
sir.
very tricky situation when we were trying with working with normal phase chromatography. Like for a chemist, normal phase chromatography is very good because in chemistry, we do not use water as a solvent for doing any kind of reactions. We use all dry solvents, right?
Okay, sir.
So what we did
Okay, sir.
actually, that there was a tricky situation that what to do. I want to use water as a mobile phase for biological samples. So for that, what was done, that the polarity of the stationary phase was changed from polar to non-polar, from polar to
Non-polar.
non-polar. And nowadays, we have the modified phases where like on silica gel itself, we did the carbon loading. We did the carbon loading.
Carbon
If we
loading.
attach a carbon chain of say 6, 8 or 18 carbon, now it will become polar or non-polar. You have a carbon chains.
Hmm.
CH, 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 CH.
Sir, it will become non-polar. Yes, sir.
Very good. So it
It
will
will
be,
become
it became
non-polar.
non-polar and water can be used as a mobile phase very easily on this situation. And we
Yes,
can
sir. Yes,
separate
sir.
molecules like protein. And you'll be surprised to know that nowadays, most of the chromatography for protein, what we are doing, it is on the reverse phase column only. It is on the reverse phase column only.
Reverse
All
phase.
HPLC,
Okay, sir.
like you might have used HPLC, you said that you have used HPLC. Which column
Yes,
you
sir.
used in your HPLC? Can you name it? You said na you used HPLC earlier.
Yes.
So what
Yes, sir.
was the
Yes, sir.
column?
Sir, it was.
And what was the mobile phase?
Hello.
What was the
Hello. Yes, sir.
what
Sir,
was your mobile phase?
the mobile phase was water and
Hmm.
acetonitrile.
Very good. You might have used say 50% water, 50% acetonitrile.
Yes, sir.
Maybe
Yes, sir.
70% water, 30% acetonitrile, like this.
Yes, sir.
Now,
Yes,
what
sir.
was the
Yes, sir.
column if you remember?
Column.
Hmm. Column you might have used, either it is known as ODS column, if you remember
Okay,
the
sir.
name ODS, or maybe like C18 you remember, C18 column.
Yes, sir. Yes, sir. The
C,
C18 column. Yes, sir.
C18 column. So C18 column is a non-polar column with C18 means 18 carbon chain. 18 means
Yes, sir.
18 carbon chain.
18 carbon chains.
So
Okay,
18
sir.
carbon chains
Yes,
means
sir.
CH2, 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 18 carbons. So it
Yes,
becomes
sir. Yes, sir.
polar or non-polar?
It becomes non-polar.
It becomes non-polar, and with that non-polar, we can use water as a solvent. And since water
Yes,
is the
sir.
major part of the mobile phase, we can easily separate our proteins. Now it helped us in two ways. One is the cost of the chromatography, it went down significantly. Why? Because the cost in chromatography is the mobile phase, cost of mobile phase.
Okay. Yes,
You
sir.
might have used, like if you have used an HPLC solvent, the HPLC solvents are
Yes,
very
sir.
costly. Say
Yes,
uh,
sir.
2000 rupees per liter, 3000 rupees per liter. Acetonitrile is very costly, around say
Yes, sir.
around 4 to 5,000 rupees per liter. Now you
Yes, sir.
see how much costly it is. But if
Yes,
my
sir.
70-80% is water, now what is the
Water.
cost
Yes.
of water per liter? Hmm? Like if you have a system in your lab itself, then the cost of water
Yes,
will be
sir.
1 rupee or 2 rupee per liter.
Yes, sir. Yes, sir.
So that
Yes, sir.
means if you run the HPLC for the whole day and
Yes,
then
sir.
you do your separation, the cost of your separation will be very low and you can make it efficiently. Okay.
Yes, sir.
So
Yes, sir.
nowadays, even for organic compounds, instead of using a normal phase chromatography, we prefer to use reverse phase chromatography because the
Okay,
cost
sir.
of separation becomes very low. Okay.
Yes, sir.
Okay. Hope you understood it.
Yes, sir.
Now the next classification is based on the operating pressure. So sometimes the chromatographies are performed on atmospheric pressure, like the way Michael Sweat did it, or it may be under pressure, like HPLC is a pressure chromatography, or GC is
High
always
pressure.
a
Yes, sir.
pressure chromatography.
Pressure.
Okay.
Yes, sir.
Now, uh, now these are like different kind of chromatographies I have given in this presentation. So what I want that you just go through the presentation, and our next class is on 10th.
Yes, sir.
So on 10th, I want that you should come up with a lot of questions. You'll go through the presentation, come up with a lot of questions, and we will discuss it on that day.
Okay, sir.
Okay, and now I think with this basic background, you'll be able to understand chromatography very easily.
Yes, sir.
Okay, and try to
Okay, sir.
understand like these these presentations I have prepared with taking input from many books, and you'll find this
Yes, sir.
presentation very interesting and very easy to understand.
Okay, sir.
Okay, good. Now you read it,
Okay, sir.
and in the next class, I will ask you a lot of questions
Okay, sir.
because I think you will be able to read it properly. You'll be able to understand it, and then I'll ask you questions, and during our discussion, you will be able to understand it very clearly.
Okay, sir. Yes, sir.
Okay, then.
Okay. Okay. Thank you, sir.
Mm-hmm.